Yeah, good morning, everybody. Yeah, my talk is on enteric viruses in chicken enteritis. Okay, I will cover my talk in under these points. First is introduction, clinical findings, necropsy findings, present situation, economic losses, and future research. If you look at what enteritis is, it is an infectious multifactorial disease of young birds and is characterized by diarrhea, depression, lethargy, and there can be mortality and there may not or may be, with gross lesions of pale intestines or ceca with watery contents. In Midwest area in the US, enteritis was recognized as one of the major problems after 2000, and most of the focus was to identify the agents that were associated with enteritis. In chicken, there are different synonyms for enteritis like runting stunting syndrome, maldigestion syndrome, malabsorption syndrome, infectious stunting syndrome, helicopter syndrome, whereas in turkeys, different names have been given like Polt enteritis complex, Polt enteritis syndrome, Polt enteritis mortality syndrome, and light turkey syndrome. It's only this Polt enteritis mortality syndrome, particularly the disease has been identified in North Carolina, where there is involvement of coronavirus and mortality is comparatively higher. As I said, disease is multifactorial. There are many agents which are associated. There can be involvement of bacteria like Salmonella, E. coli, Enterococcus, or Clostridium, or there can be involvement of protozoa, and there can be involvement of viruses. So in my talk, I'll concentrate on viruses. Different viruses of both classes, RNA and DNA, are involved. Among the RNA viruses, primarily the viruses are rotavirus, reovirus, astrovirus, picornavirus, and picorna-like virus, whereas DNA virus, they are adenovirus and parvovirus. Reoviruses, majority of the reoviruses are non-pathogenic except few which may cause lameness in birds, but the reoviruses of chicken origin are different from reoviruses of turkey origin. Among rotaviruses, I'm discussing group 2, group A and group D. Group A can cause disease in mammals and birds, whereas group D is exclusive to birds, and both the groups have been identified in poultry suffering from enteritis. Astroviruses, this group has gained more attention recently because astroviruses of chicken origin differ a lot from astroviruses of turkey origin. In chicken, avian nephritis virus and chicken astrovirus have been identified, whereas in turkeys, it is turkey astrovirus 1 and turkey astrovirus 2. Picornavirus, adenovirus, and parvovirus, recently in last 3 or 4 years, these viruses have been identified to cause enteritis in birds. Now, majority are RNA viruses, as I told, like rotavirus, astro, or reo. These viruses are unstable. There can be mutation. there can be recombination. Even we have observed recombination in reoviruses, and due to that, there can be different genotypes and serotypes. And due to these virus variations, there can be difference in antigenicity of the virus, and there can be difference in pathogenicity of the virus, and that's why under field condition we observe that certain viruses can cause more severe disease, whereas certain viruses are not able to cause severe disease and cause only mild disease. There can be problems with diagnosis, with epidemiology, and the vaccines. Coming to the clinical presentation, as I told, feces are watery, there can be paste event, there is poor growth of the bird, and it is the primary reason for economic losses to the poultry growers throughout the world. There can be lameness in some of the affected birds, and there can be mortality, and mortality is more where coronavirus involvement is there. These are all pictures of paste event, this one, and if you just look at this place, these are all direct feces. Coming to the necropsy findings, as I've told, the intestines become thin-walled. If you just compare the intestine like here and here, you'll find these are the pale intestines, and this is the cecal contents become watery and frothy. If you look at the cecal contents, these are all watery and frothy. In duodenum, because duodenum and jejunum, these are the two portions of the gut where majority of the nutrients are absorbed. And if gut is not healthy, birds are not able to gain body weight. In duodenum, there is thickness of wall, or there can be paleness of wall with undigested material, and this material body is not able to absorb the nutrients. Like there can be arrhythmia of the villi, and due to which the absorption capacity of the villi is reduced. Similarly, in jejunum, you will find the watery contents with undigested feed and increased mucus. Various studies have been carried out to detect which are the important viruses that are present in turkeys and chicken in different areas. In the US, studies have been conducted which indicate that chicken astrovirus is present in 86% of the samples from enteric cases that were collected in certain parts. In another study, astroviruses were again detected in 86% of the flocks. We also conducted a study in Midwest where we collected samples from 151 flocks, and of these flocks, different agents were detected either by cultural examination, particularly for bacteria, and viruses were detected based on electron microscopy. Then we conducted another study to determine whether the viruses detected by electron microscopy are the same or they are different. In the sense, if we go to previous slide, there are certain cases where we detected small round virus, and when we opted for PCR, we found that these small round viruses were actually turkey astroviruses. And in no case we could detect reovirus by electron microscopy, but we could detect 40% of cases as positive for reovirus by this PCR. Then we conducted another study to determine whether these viruses are present alone or in combination, and we found that in 8 cases, only one virus was present, whereas in 20 cases, 2 viruses were present, and in 15 cases, 3 viruses were present, and 3 viruses were rotavirus, astrovirus, and reovirus. Studies have been conducted in Korea, where they tested 34 commercial frogs for presence of enteric viruses, and the viruses were detected in 85.3% of the flocks. Similarly, studies were conducted in Brazil, and again the samples were found positive for individual virus and more than 2 viruses. Then we also conducted studies in India, where we collected samples from 65 flocks. What we did, from each flock, we collected samples from 5 to 10 birds, and all samples were pooled, and those pooled samples were tested by RT-PCR for different enteric viruses. Surprisingly, none of the sample was positive for rotavirus and reovirus, whereas samples were positive for chicken astrovirus alone, avian nephritis virus alone, and in combination. You can say 31 samples were positive for chicken astrovirus, 4 for avian nephritis virus, whereas 26 samples were positive for both viruses, and 93% of the samples were positive for these 2 viruses alone or in combination. Then we sequenced some of the positive PCR products. We use this polymerase gene because this is the conserved gene of among the astroviruses, and within even using the conserved gene, we could found that taking all astroviruses into consideration, that avian astroviruses and within avian chicken astroviruses group under 3 groups, group 1, group 2, and group 3. All Indian strains were in group 1. As I told, polymerase gene is the conserved gene. Even in conserved gene, there were changes of amino acid, let's say from isoleucine to valine, and 9 of the 12 strains showed this change. Similarly, you will find many strains revealed changes at the amino acid level. The impact we don't know at present, but studies are required to be undertaken or to be conducted to determine whether these changes affect the antigenicity or pathogenicity of the virus. Similarly, we analyzed certain sequences of avian nephritis virus, and we found all this, particularly this group, it is avian nephritis virus 1, and this is another group of Turkey origin, this nephritis virus, which comes under group 2. We also got amino acid changes in the conserved gene in avian nephritis virus, and we are conducting studies to determine the changes in capsid gene, which is more variable. Then we undertook another study to determine to what extent the viral load in birds is, or up to which age birds harbor the enteric viruses. That study was conducted in Midwest, where we selected 5 breeder flocks and we monitored these flocks for 9 weeks, and we collected samples at regular intervals from these flocks, and we observed that viral load is maximum up to 3 weeks of age, starting from day 0 of age. Now this is very important, means the losses which occur due to enteritis or which are thought to be occurring due to enteritis, according to a report, the estimated losses only from growth depression due to poult enteritis are between $300 to $400 million annually if we consider 10% to 15% growth depression throughout the turkey industry in the US. Later on, we conducted experimental studies. 5 studies were undertaken using different inoculum from enteritis birds, and we observed that there is growth depression up to 31%. Another study was undertaken not by us, where the study was undertaken only in one farm with 2 or 3 flocks of 20,000 birds, and enteritis resulted in 12% reduction in body weight and 10.9% increase in feed conversion ratio compared to healthy birds. And final body weight of the birds in these flocks ranged from 4.63 to 7.94 pounds, and losses to the producer ranged from $878 to $1,480 in just one flock. This is of the only one farm, and there was increased feed cost that ranged from $370 to $739 per flock. So we can think of to what extent enteritis is causing losses to the poultry producer. Coming to the scenario in India, where the enteritis is also a major problem, we are the second largest egg producer in the world and third largest broiler chicken meat producer as in 2015, and market is estimated to be worth rupees $900 billion, and by 2016 we expect meat production, chicken meat production to up by 8%. And enteritis is a major problem responsible for reduced weight in birds, particularly at marketing age. We are conducting experimental studies in India to determine whether it's astrovirus or avian nephritis virus which is responsible for causing enteritis, or there is association of another pathogens. We presume that it's the enteric viruses which initiate this disease process, and later on it's taken up by bacteria and protozoa. Though surveillance studies are required, we conducted this study only in a limited area. More studies are required to understand the exact nature and prevalence of viruses in different parts of the country. Next generation sequencing studies are also required, and this aspect need to be undertaken, that what are the interaction of different pathogens that goes on inside the body, where different pathogens they interact and may help to increase the disease causing capability of the pathogen. Economic losses, yes, these are required, and on these basis we can think of what preventive and control measures can be taken. I acknowledge the help and guidance from Dr. Goel, Professor Ziegler, Dr. Patnaik, Sunil Moore, Babita, and Pawan. Thank you. Question, please.